Good morning. So today we will start the new chapter, that is your cell biology or cytology. This is a branch of biology in which we study about the cells, right? Now, the morphological, biochemical, physiological and developmentally evolutionary study of this cell is known as cytology. So what I said, the morphological, biochemical, physiological and developmental. Right? So the morphological, biochemical, physiological, developmental and evolutionary study of cell is included under your, right? Now what is cell? What is cell? Cell is the structural and functional unit of life that is delimited by the semi-permeable membrane. A semi-permeable membrane. So this is the structural and functional unit of life that is delimited by the semi-permeable membrane and is capable of self-reproduction, right? And a medium that is free of other living subsystem. So it is capable of self-reproduction in a medium which is free of the other system, right? Now, this cell, this was first discovered by Robert Hooke, right? So Robert Hooke, he discovered the cell, right? And the cell theory was given by Schleiden and Swan. Cell theory was given by Schleiden and Swan, right? So it was discovered by Robert Hooke and the theory, cell theory was given by Schleiden and Swan. Schleiden and Swan actually said that all tissues are composed of the cells and that the body's metabolism is the result of your this cellular activity, that is independently, right? Then Virchow also said that he also gave this cell theory and said that Virchow said omnis cellula celluli. That means the cell arises from the pre-existing cells, right? So the Virchow's theory was that he said omnis cellula celluli. That means this arises from the pre-existing cell. So this is just the introduction about the cell. What is cell? What is the cell theory? Who gave this? And what is the theory of Virchow? Right? Theory of Virchow is very interesting and important because this is the basic of our every physiological phenomenon, that is the cell division, right? Now here all the living organisms, they are not cellular, but some are acellular also. So what are the acellular living organisms? Like the bacteria, algae, right? So this I am saying that these are the acellular organisms, right? Because they actually do not exhibit the typical or a true cellular organization, right? That is, you will see, I will tell you later on, that you will see that is bacteria and algae, they do not contain the nuclear material, and their nuclear material is actually devoid of any envelope, that is the nuclear membrane is not present. So they are included under prokaryotes. They are included and the prokaryotes, right? Now, the next important thing is about protoplasm. Protoplasm is the next important part of the cell. Now, this protoplasm term was coined by Purkinje. This was coined by Purkinje, right? Now, this protoplasm occurs in all cells. The partition between the plasma membrane and nucleus is the cytoplasm, and this occurs, that part that occurs in the nucleus is the nucleoplasm. So, what is cytoplasm and what is nucleoplasm is clear? The protoplasm or the portion that is found between this plasma membrane and nucleus. This is found between plasma membrane and nucleus, right? So this is the cytoplasm, and the nucleoplasm is that occurs in the nucleus, right? Now, so in this way you can say that this cytoplasm and nucleoplasm, they constitute your protoplasm, right? So, the cell is a mass protoplasm having a distinct nucleus and is limited by a plasma membrane, right? Then, let's see what are the types of this cell. Right? The cells, they are of two types, the prokaryotes or the prokaryotic cell and the eukaryotic cell. Right? The prokaryotic cell and the eukaryotic cell. This are known as prokaryotes and these are eukaryotes. Right? So, let's see first the prokaryotes. Just I told you that the bacteria and algae, they are included under this prokaryotes. They are not having the typical cellular organization. That is why they are considered as acellular, right? Now, I told you that this is devoid of the nuclear membrane. That means this lacks the true nucleus. This lacks the true nucleus. If I am saying that this is lacking the true nucleus, that means there is only one or two molecule of DNA or RNA is present, right? So in this only one or two DNA or RNA molecules are present. They are not having any nuclear membrane, right? Now, this prokaryotes, they have the rigid cell wall that is made up of, cell wall is made up of your amino acids and carbohydrate. Amino acid and carbohydrate. Right? So the cell wall is made up of the amino acid carbohydrate and it is rigid. Right? Now, this prokaryotes, the cytoplasm do not have any flow. Cytoplasm do not have any flow. Right? That is no cytosis movement. Right? Now, this protein in this nucleus, you know, the nucleus is made up of DNA and protein, right? So, this protein, nuclear protein that contains histone in the eukaryotes, this is absent in the prokaryotes, right? Then it also does not contain some cellular organelles like your mitochondria, the cellular organelles like mitochondria, chloroplast and Golgi bodies, they are absent, right? And as there is no nuclear membrane and therefore their nucleus does not show any division, right? No division in the nucleus, right? And the bacteria example is your bacteria and cyanobacteria, that we also call blue-gill algae, right? We also call it blue-green algae, or in short we write this BGA. Okay, now we will see the eukaryotes. These eukaryotes, they are the cells of higher plants, right? This prokaryotic or the prokaryote cells, they are the primitive cells. Remember, they are the primitive cells. From the evolutionary point of view, these prokaryotes are the primitive organisms, right? Now, the next, let's see the eukaryotes. These eukaryotes, they have the cells of higher plants and animals. They have well-developed nuclear membrane, right? Well-developed nuclear membrane is present. Then the organelles, all organelles are present here, right? So with this introduction, let's see first. See, I had told you this prokaryote and eukaryote division, right? Now I would like to tell you here only something about the viruses, right? So this virus, this is the obligate parasite, as you all know. Now this is in fact prokaryote and eukaryote both, right? This lacks the cellular organization, contains some genetic material that is only by the macromolecular organization, genetic material and characteristic mode of inheritance is found here. This is not having its own metabolic machinery, right? Rather, when it attacks the host, it hijacks the cellular metabolism or the metabolic machinery of the host, right? You might have heard this name, tobacco mosaic virus, TMV, or the tobacco mosaic virus. Right? So this very important. Remember, this was first identified or discovered by W. Stanley, right? And these viruses, they are very small, that is, they are submicroscopic entity. Remember, they are the submicroscopic entity. Okay? The size varies from 10 to 250 millimicron. Okay? Now, mostly these viruses are smaller than the bacteria, but there is one virus which is having big size than the bacteria, that is your Cytology virus. The Cytology virus is having the size of 0.75 millimicron, and the size of, you might have heard the name of PPLO, right, or mycoplasma. This is the size of 25 micrometer diameter. Now, these viruses, they are divided depending upon the type of host, depending upon their genetic material, and depending upon their metabolism, right? So, according to the type of host, if we divide them, then they can be a plant virus or the animal virus. Okay? Then, according to the genetic material, they are RNA virus and DNA virus. This is on the basis of genetic material, and this is on the basis of host. Right? Now, this RNA virus means they are having the genetic material RNA, and DNA virus, they are having genetic material DNA. Right? This RNA virus, they are again retrovirus. Here, this virus carry the RNA as the genetic material, and they are mainly they are having the single-stranded or the double-stranded. They can be single-stranded or double-stranded. So, if you are asked to give the example of single-stranded RNA virus, then you will write the tobacco mosaic virus and polio virus. But the double-stranded RNA virus, the example is the rice dwarf virus. Right? This example you must know this, right? Now let's see the DNA virus. This DNA virus, just I told you, the DNA is the genetic material, right? And this can be again single-stranded or double-stranded. The example of the single-stranded DNA virus is the 5174 virus, right? Its name is 5174 virus. And the example of this double-stranded DNA virus, they are here mostly the bacteriophages are included. Bacteriophages, right? The most common animal and the animal viruses. The animal viruses are also this DNA virus, right? Now, mostly the animal viruses are the DNA virus. Remember this point. Okay. Now, here I told you bacteriophage. Regarding this bacteriophage, I will try to give you some more information. That there is T4 bacteriophage, right? This is very common, very important. This actually infects the colon bacillus, that is the E. coli, right? Bacteriophage, they are the viruses which infect the bacteria, right? So this T4 bacteriophage, this infects the colon bacillus E. coli, right? And this has a, let me show you the diagram. So this bacteriophage, that is I am telling you the T4 bacteriophage, this is mainly, I told you, this mainly infects the E. coli bacillus, right, or the Escherichia coli bacillus. Now, they have a polyhedral body and a short neck, right, and then the collar, that is straight, and the genetic material is the DNA virus, right, and this is similar, right. Now, here this remains tightly packed within this protein capsule, that is called your capsid. This is called capsid. Then this has a short neck. This has a short neck, and this contains the collar connects with this head with this collar. This neck is the connecting system, right? This connects the head with the tail, right? Now this tail, this is the tail. Tail, this consists of which is having the phages microphone, right? Now this hexagonal end plate. This is having a hexagonal end plate. This has 6 slender fibers. You can see here 1, 2, 3, 4, 5, 6. These are the 6 slender fibers, and this 6 short spikes that actually protrude from the end plate, and this helps in inhibition. When it gets attached over the bacterial cell wall, this helps in penetration, right? Now, mode of infection of this T4 bacteriophage is highly unrecognized, right? It is very mechanized. The mode of infection of this T4 bacteriophage is highly mechanized. So what happens at the time of this infection? of this bacteria, this get anchored to this bacterial wall by this cell fibers. Then the enzyme lysozyme is secreted by these cell fibers, right? And a part of bacterial envelope is here dissolved, right? And finally the phage DNA is injected inside, right? And the capsid that remains outside. Right? And the DNA has come inside. Okay? So the viral DNA now here hijacks this bacterial cell machinery and directs the synthesis of viral protein. Remember, these viruses are very clever, right? So they hijack the metabolic machinery of the bacteria, and then it instructs or directs the synthesis of viral protein, or the viral, you can say that this viral DNA is incorporated into the bacterial genome, and then this replicate along with it. Okay. Now, next we will see another small organism that I had taken the name, that is the PPLO or mycoplasma. PPLO or mycoplasma. PPLO means pleuropneumonia-like organism. Pleuropneumonia-like organism. Or we also call it PPLO or mycoplasma. Right? Now, these are the bacteria-like organisms, but it differs from bacteria in the point that this lacks the cell wall and mesosome. Right? So, this does not contain the cell wall and mesosome. Right? Now, here they are very small, or you can say that they are smaller than the bacteria. The most widely structural genome of this PPLO or is the mycoplasma. What I am saying, the most widely structural genus of this PPLO is the mycoplasma. Now, this is the smaller organism that can be grown on the cell-free media. Remember. Now, this they are some are saprophytic, and some are parasitic. So here you note that if you are asked to give the example of parasitic PPLO or parasitic mycoplasma, then the example will be your Mycoplasma gallisepticum. Right? But if you are asked to give an example of saprophytic, then saprophytic mycoplasma, then the example you will write the Mycoplasma leglavi. Right? So this example you must remember, right? So what I said, that this pleuropneumonia-like organism, they have this cell membrane, they are having the DNA material, and here the DNA is circular. DNA is circular DNA, right? The ribosomes, granules, vacuoles, they are also found in the cytoplasm. Now, the next microorganism that I will tell you is the chlamydia. Chlamydia and rickettsia, they are the small bacteria, and they are parasitic, and here the disease that are mainly caused by this chlamydia and rickettsia are the parrot fever, Q fever, typhus fever. Remember, in man the typhus fever is caused by this, right? Here the cell wall is present and this reproduces like bacteria. The genetic material, they lie naked in the cytoplasm. only this much of information regarding this chlamydia and rickettsia is sufficient for you. Then we will see the bacteria. Right? Here the bacteria, they are the microscopic structures and they are unicellular, prokaryotic, autotrophic, saprophytic and parasitic. Remember, the bacteria are having every mode of their nutrition, right? Autotrophic, saprophytic and parasitic. Now, the structure, you know that the bacteria is having the cell wall. The cell wall is made up of polysaccharide, right? Then they are also, this is actually this polysaccharide, they are having the capsule around the cell wall. That is the capsule. It is made up of polysaccharide, right? Then inner to this capsule there is cell membrane, and this cell wall, as I told you initially when I was telling you about prokaryote, that this is made up of the amino acids and carbohydrate, right? So this is made up of your proteins, then the inorganic salts, right, and amino acids. Here the diamino palmilic acid, and muramic acid. Remember, this is found in the cell wall, right? Now, then the third is the plasma membrane. This plasma membrane, here the cytoplasm is actually derivated by the thin membrane of lipid and protein, right? And they are called, this is known as the plasma membrane. So the plasma membrane of the bacterial cell, they may form a multi-layered structure, and that is called mesosome. So the plasma membrane of the bacteria that forms a multi-layered structure, that is called mesosome, right? This mesosome performs very important functions, right? The function of mesosome: respiratory function. First, the respiratory function. This has the respiratory enzymes, right? The respiratory enzymes are concentrated over this mesosomes, so this helps in respiratory function. Then this is also having the secretory function. This secretes many important things, right? Then it also takes part in the duplication of chromosome, or you can say chromosomal duplication. This helps in chromosomal duplication also, right? Then that means when there is duplication of the daughter and the formation of the daughter chromosome to each daughter cell, this helps, right? Now they are also found, these mesosomes are also found associated with the DNA. So this is thought that it is the site of DNA replication system also, right? So this is also thought to be the site of DNA replication system. Okay? Then we will see the cytoplasm. Inner to the plasma membrane is the cytoplasm, right? This cytoplasm of the bacterial cell is very dense, colloidal, and this contains the granules of, this is colloidal, dense, and contains the granules of glycogen and fat. Remember, this cytoplasm is containing glycogen, protein and fat granules, right? The endoplasmic reticulum, Golgi body, lysosome, centriole, all are found. Then bacteria, remember one thing, that this bacteria contain free ribosome. Okay. So the bacterial cell has free ribosome. The genetic information of this bacterial cell is carried by the DNA molecule, and that DNA we call here bacterial chromosome or genome 4. Genome 4 or bacterial chromosome. Right? But I am saying the genetic information of each bacterial cell is carried by the DNA molecule that is known as genome 4 or bacterial chromosome. This DNA is present in the distinct nuclear region that is known as nucleoid. Right? So the photosynthetic bacteria that contains the bacteriochlorophyll. I told you that the bacteria can be autotrophic, means they are photosynthetic, right? So if they are photosynthetic, then they contain the photosynthetic pigment that is named bacteriochlorophyll, right? Bacteriochlorophyll and other pigments like phycoerythrin, etc. Right? Now the photosynthetic pigment and this enzyme, they are associated with the internal membrane, and they are arranged as the lamellae, tubercle or vesicles, and they form the chromatophores. Right? Now, this bacteria also possess flagella. This bacteria also possess flagella that can be one to many. Right? This flagellum, they arise from the basal body. This flagellum, this arises from the basal body. They are all basal granule and is made up of a protein that is called flagellin. Remember, the name of protein that is forming this flagella is the flagellin, and this is originating from the basal granule, rather, you remember. Yes, basal granule. Now, the cells of some bacteria, they also have hair-like structure which is extracellular, and this is known as pili, right? And this is known as the pili. So what is pili? This pili is the extracellular hair-like structure, right? That is present as a cellular outgrowth. Actually, this is an outgrowth, right? Now, what is the significance of this pili? Here, you remember that in male bacteria of certain strains, they contain this special hollow sex villi, right? They are hollow, right? And then actually form a bridge that is known as conjugation bridge during the reproduction, right? This one, they get attached with this, and this forms a conjugation canal or conjugation bridge. Okay. Now, this pili in this pathogenic bacteria, they also recognize the host, right? They are actually composed of the subunits. They are mainly composed of various subunits, and here you remember that this is mainly concerned with this reproduction, right? And they are, now let's see the structure. They are actually the helically arranged subunit. Helically arranged subunit that is made up of protein and is known as pyline. Pyline. Okay. So now many, I have told you that they are mainly made up of this helically arranged subunit that are known as pyline. With this information about the, and I think this is sufficient for you, right? This is a prokaryotic cell is now over. Now let's see the eukaryotic cell. Eukaryotic cell, right? We will see the structure of the eukaryotic cell. Right? Now here there are two envelope system and is larger than the prokaryotes. Remember this point. And the secondary membrane, that envelope, is the nucleus and other cellular organelle. occurs in the plant, that is algae to angiosperm, and in animals, that is protozoan to mammals. Right? Now the structure of the cell we will see. The outer covering is the cell wall and is non-living. Remember, this is a non-living covering and is secreted by the cell itself. Okay, and is mainly composed of the polysaccharide carbohydrate cellulose. Right? Cellulose. And this is present in the plants only. Right? This provides protection and support to the plasma membrane and cytoplasm. Okay. There are some fits in this through which the cytoplasmic strands they project to the adjacent cell, and that we call plasmodesmata. Plasmodesmata. Okay. Now the next structure is the plasma membrane. Remember, the cell wall is present only in the plants, right? But this is absent in the animal cell, right? Now the plasma membrane, this is present in both plant cell and animal cell. The outer covering that is present in the animal as well as plant is the plasma membrane. This is also known as cell membrane. Now here you do not get confused with the cell membrane and cell wall, right? This is cell membrane, plasma membrane, cell membrane, or this is also called plasma lemma. Okay. This is living. This is living. This is non-living, right? Cell wall was non-living, and this is living, elastic, and semi-permeable or selectively permeable. Semi-permeable. Right? Now, this actually delimits the protoplasm from exterior. Then it regulates the movement of the material from the inside or outside the cells. Right? So, this is the lipoprotein complex. This is made up of lipoprotein complex, and this contains actually the small pores. The size of this pore is about 8 angstrom, and through this the molecules may exhibit free movement. Right? Then the next is your cytoplasm. Cytoplasm. This is the external form that this actually extends from the plasma membrane to nuclear membrane, and this is consisting of the cytoplasmic matrix and cytoplasmic structures. What I said, this we will study into two parts: cytoplasm matrix and cytoplasm organelles, right, or structures. Okay, with this I would first like to tell you in detail about the plasma membrane. I told you that this plasma membrane is present in the animal cell as well as in the plant cell, and this is living, right? And this elastic made up of lipoprotein complex. The structure and function, let's first see the structure and function of this plasma membrane. This plasma membrane term was mainly first coined by Nageli and Kremer. This term was coined by Nageli and Kremer, right? And this plasma membrane lies between the cell wall and cytoplasm. You have seen it, right? Now, the thickness of this plasma membrane, this extends about 70 to 100 angstrom, and is made up, it is lipoprotein complex. So, this is made up of mainly the lipid and protein, and some amount of polysaccharide is also found. mostly protein. This is mostly protein, right? Mostly protein part predominates except the myelin sheath. Remember, myelin sheath. In myelin sheath, the lipid predominates. Remember. But I am saying in plasma membrane, the lipid part mainly predominates, protein part mainly predominates, but in the myelin sheath cells, this lipid is mainly predominating. Now, there are different scientists who have given their views regarding this structure of the plasma membrane. In this, we will take the few important theories, that is the lipid bilayer theory, right? The lipid bilayer theory, or you can say the lipid bilayer model. Okay? Lipid bilayer theory or lipid bilayer model, and this was given by Gorter and Gandel. We must remember this name of the scientist who had given this theory, right? Now, so this was given by Gorter and Gandel. The lipid is arranged here in the bilayer of oppositely oriented monomolecular layers, right? Now, there are hydrophobic hydrocarbons that end in the molecule that are faces inwards and towards each other, like this. At the hydrophobic ends, they are actually facing inwards and towards each other. This is the hydrophobic end, right? Sorry. And the hydrophilic ends, or the polar layer, they face outwards toward the aqueous phase, right? Now, this arrangement is the essence of the bimolecular leaflet or lipid bimolecular model or membrane structure. Then, the second type of model or the theory was given by Danieli-Davison model. Danieli-Davison model. You remember this, only you have to remember that the hydrophobic ends, they are facing inwards and they are directed inside, right? And the hydrophilic or the polar towards outside. And the name here, right? Then the Danieli-Davison method model, here the double layer is formed, which is of phospholipid. He said that this is of phospholipid end structure, right? Now, in addition to this polar ends of the lipid molecules, they are also associated with some monomolecular layer of globular protein. So, the lipid molecule, they are actually entered between the continuous layer of the protein, right? So, the lipid molecules, they are you can say that they are actually sandwiched, right? These lipid molecules are sandwiched between the continuous layer of protein. Right? So this is the continuous layer of protein, and this lipid molecule is sandwiched. Okay. Now the third is the unit membrane model. This unit membrane model, this was given by Robertson. He said that this is trilaminar. Right? So here the two dark electron dense lines, they are separated by a light region. In this way, this is trilaminar. Two dark lines, right? They are separated by the light region. Therefore, this is trilaminar. Now, here a dense band. What I am saying, there are two dense bands, right? So, this dense band is made up of your protein and polar group of lipid. Protein and polar group of lipid. So the dense band is made up of protein and polar group of the lipids, right? And the light zone or the clear zone, this consists of bimolecular protein layer. Bimolecular protein layer. Okay. So this consists of bimolecular protein layer without polar group, right? Now this protein layer is an extended B-sheet form. Remember, extended, here the protein layer is of extended B-sheet form, right? Whereas in that Daniel-Davidson model, that protein was globular. This is the difference, remember. Here, this is extended B-sheet form protein layer, whereas in Daniel-Davidson model, this was globular, right? Then the next model was greater membrane model. Greater membrane model. So let's see what is this greater membrane model. Here the internal surface of the membrane is thought to be covered by the unconjugated protein and the outer is having glycoprotein. But I am saying the inner one is unconjugated protein and the outer surface is made up of glycoprotein. Okay, now here these are mainly composed of the structural protein, that is the inner one, the unconjugated protein, and the outer surface is glycoprotein. Now attached to this glycoprotein, the oligosaccharide side chains are there, and they have negatively charged acid terminals, right? Okay, now so you can say that the membrane is asymmetrical and this has the structural polarity. Okay. Now we will see the most important model, that is the fluid mosaic model. Fluid mosaic model. This fluid mosaic model was given by Singer and Nicholson. Here the lipid and proteins, they are arranged in the mosaic pattern, right? They also said that this is made up of lipid and protein and they are arranged in the mosaic pattern. Now, continuous bilayer of this phospholipid molecule, in this the proteins are embedded. You might have seen the mosaic, right? In this small, the white protein, you have seen the mosaic pattern, right? So in this the cement base is there and some small pieces of the marble, they are embedded in this, right? So this is the mosaic, right? In this way here also, what happens? There is continuous layer of the phospholipid. Remember this thing, that the phospholipid is making the continuous layer, and in this the proteins are embedded. Okay, within this lipid bilayer the proteins are embedded. That means they are not fixed. These proteins are not fixed within the lipid bilayer, but that they are free to move, right? Now this lipid protein association is actually hydrophobic, and this accounts for the fluidity of the membrane. This accounts for the fluidity of the membrane, right? Now these proteins, whatever is found in this membrane, they are of two types, the extrinsic and intrinsic. So this extrinsic or intrinsic. Extrinsic is peripheral. This also you can say the peripheral proteins, and intrinsic they are the integral proteins. Right? So these proteins of this plasma membrane, they are of two types, extrinsic they are peripheral and intrinsic they are integral. Now the peripheral proteins, they are actually readily dissociate from the membrane and they are insoluble and they lie outside the lipid bilayer. Extrinsic means outside the lipid bilayer, right? And the integral one or the intrinsic protein, they are relatively insoluble and they dissociate easily with the they do not dissociate easily, but they dissociate with difficulty, right? Now some of this may penetrate either surface of the lipid bilayer, and when they are penetrating the others they penetrate, but I am saying some of these proteins they may partially penetrate and some of them penetrate completely, right? Now many of these proteins, they are the enzymes, and these enzymes, that is permease. You know, this permease is responsible for permeability, so and this therefore helps in the entry of the substance, right? Entry of substance they help in, right? Now what is the significance of this model? This has incorporated, they say that this incorporates the larger area of uninterrupted phospholipid layer, which provides the permeability barrier, right? So what I am saying, the continuous layer of phospholipid is providing the permeability barrier, right? Then the globular protein, this is responsible for many, this can control for the many metabolic activities, right? Okay. Then this permease that has that was responsible for the entry of the substances, right? Then the fluidity of membrane, I have told you that this fluidity of membrane is due to the free moving movement of that protein particles, right? Now we will see some spatial structure of the plasma membrane. So this structure of the plasma membrane, that are different theories that are given by different scientists is clear. Okay. So with this we will now start with some specialized structure of plasma membrane. This specialized structure of the plasma membrane. Now let's see what are the structures. See, you know, this plasma membrane is mainly responsible for your absorption, secretion, transportation, and these are all performed by the cell surface of this cells, right? And for this some modifications actually occur, right? You might have heard this name, microvilli, isn't it? Microvilli. These are the columnar epithelial cells. These are the columnar epithelial cells of the internal epithelium, and they perform the active absorption of the digested food, right? They are very finger-like, right? Like this. They are the finger-like structure and they are lined by columnar epithelial cells. Okay, so this finger-like structures or the cytoplasmic process that are known as the microvilli, they actually put forth from the free surface on the epithelial cells, and each microvilli size is about 0.6 to 0.8 micrometer and in length, right? 0.6 to 0.8 micrometer in length and 1 micrometer diameter. Okay. Now, this narrow space between this microvilli that actually forms a type of sieve through which the substance can pass easily through the process of absorption. These microvilli are also present in the hepatic cells, then mesothelial cells of the convoluted tubule of the kidney, and epithelial cells of the gallbladder, uterus, and the yolk sac. They are present in the, what I said, they are, first we have seen that they are present in the alimentary canal, right? That is mainly the intestine. Then secondly, they are also present in the hepatic cells, or you can say the hepatocytes, right? Then in kidney also, I told you that they are also present in the kidney, that is the convoluted tubule of the kidney. Convoluted tubule of kidney, right? Then they are also present in the epithelial cells of the gallbladder, uterus, and yolk sac. Right? Now, second is first was the microvilli. Second special structure is the infolding. Some infoldings in the plasma membrane. Let's see what are these infoldings of the plasma membrane. In kidney cells or the renal cells, there are many infoldings, and this infoldings mainly contains the mitochondria, which provide energy to the plasma membrane for the active transport of the substance. So these infoldings are mainly helpful in active transport of the substance, and this infolding they contain the mitochondria, right? Now, this plasma membrane, they are also specialized at some sites. So this was for the purpose of absorption, right? Now, for the purpose of contact or during modification or specialization of the plasma membrane due to contact, this plasma membrane modification due to contact, we will see now. First, this plasma membrane of the adjacent cell, they are often separated by a uniform intercellular space. But at some places, the plasma membrane, they are very much adjacent and they may come close to each other. Means there is no intercellular space, and that's why there occurs contact, right? Now, in some cases, the membranes, protein of these adjacent cells, they interact with each other in a very highly specialized way, and they provide some intercellular junctions. And these junctions are actually the devices that glue the cells, and some they act as barrier, and therefore they prevent the pass of fluid from one to the other. That is, they prevent the passage of fluid from one cell to the other, right? And this type of modification, first let's see the desmosome. Desmosome is also known as macular adherens. Macular adherens. So when the plasma membrane of the adjacent cells, they actually become very thick, and at certain places where they are thick, fine filaments that are known as tonofilament. So what I am saying, the plasma membrane at the site of contact, at some places they become thick, and from this the filament-like structure or the fine thread-like structure, that is the tonofilament, they radiate towards the interior of the cell from these thickened areas, and then they are known as the desmosomes. Now the intercellular gap, this contains this acid mucopolysaccharide and the protein and the extracellular material that provides the adhesion, right? And this tonofilament, this provides the intracellular mechanical support, and the desmosome, this is mainly for the gluing the cells together. Okay. Then second structure is the terminal bar. Terminal bars, or this is known as zonula adherens. This is macula adherens, this is zonula adherens. They are also called intermediary junction, right? And there is actually dissolution of the this structure. This mainly resembles just like this desmosome, right? But they do not have tonofrill, or you can say the tonofilaments are absent, or they lack the tonofilament, right? Then the plasma membrane, this is thickened. Just I told you that this is similar, structure is similar with the desmosome, right? This terminal bars or zonula adherens, they are present in the intermediate portion of the plasma membrane of the columnar cells, right? Then the third is the interdigitation. Interdigitation. This the plasma membrane of these adjacent cells, they actually give rise some finger-like projections. They are called interdigitation. The name itself suggests interdigitation, right? So, with this basic knowledge of this plasma membrane, now we will see in detail about the cell. I told you that this cell is the structural and functional unit of the body, right? So the three functional regions of the cell, just I told you, three functional regions of the cells are the, yes, they are the plasma membrane or cell membrane, then the nucleus and the cytoplasm, isn't it? Right? And regarding this plasma membrane, I will take in short that that will be a revision, right? Now, this is the outer covering of each cell, plant, animal, and the microorganism. All right. The cellular organelles like the mitochondria, chloroplast, lysosome, nucleus, Golgi body, every organelle, they are covered by this unit membrane. So this plasma membrane is the unit membrane, right? This is thin, delicate, elastic, and semi-permeable, isn't it? And 75% of this is made up of the phospholipid, and 25% is protein. I told you that it's mainly is made up of the phospholipid. That is the key element. Remember, the phospholipid is the key element, and 25% is made up of the protein and cholesterol. Right? And Singer Nicholson, he suggested the model that is called fluid mosaic model, and this is actually with this model, the ultrastructure of the plasma membrane is explained. Okay. With this, some few points regarding this plasma membrane, we will now see this regarding this. I will again one more thing, that there are number of protein icebergs that is floating in the sea of lipid. Remember, this is also said that there are number of protein icebergs that are floating in the sea of lipid. Right? Fluid mosaic model in this. Regarding this is one sentence is also new. Now this nuclear model is accepted because this describes the properties and all the organization of the membrane. Right? Why this was accepted is clear. Now I told you that contains the protein, right? So what is the role of these proteins? Some of them they act as enzyme. One name I told you, permease, which catalyzes the permeability reaction, right? And then there are some transport proteins which help in the movement of water soluble ions, right? Then this also act as a pump for selective transport. Then the receptor proteins are there which help in endocytosis. Endocytosis, right? You must remember a point that, rather you noted that the presence of protein and lipid, this provides flexibility to the membrane and this helps in endocytosis. Okay, so in this way, this permits the entry and exit of many materials inside the cell or outside the cell. Now, then it performs the different diffusion, osmosis, endocytosis and active transport. Here I would tell you something about this process, how they are taking place, right? Now, the first I told you diffusion, right? Diffusion. So, this process, diffusion, what is diffusion? This is the spontaneous movement of the molecule from higher concentration to lower concentration, right? And this leads to uniform concentration, right? So, this is mainly seen in the gaseous phase, right? Then the liquid phase. Now, in this, you might have seen that in unicellular organism, no special organ system is formed, right? But for this, the metabolic mechanism has to occur. So, for gaseous exchange, for respiration, the gaseous exchange has to occur. For this diffusion process is useful. The carbon dioxide comes from inside of the cell to outside and oxygen from outside to inside. So, in diffusion, the gaseous exchange is the very good example, right? Now, the next is the osmosis. Next process I told you is the osmosis. What is osmosis? This is the spontaneous movement of the water molecule through the selective permeable membrane. And I told you that the selective permeable membrane, here the plasma membrane is the selective permeable membrane. So, it helps in osmosis, right? Now, the movement of water molecule from the region of higher concentration to the region of lower concentration through the semi-permeable membrane is osmosis, right? Here, no spending of energy occurs. Remember, in osmosis, there is no loss of energy, right? So, this is purely mechanical diffusion process, right? The solutions, depending upon this osmolality, the solutions can be hypotonic, hypertonic or isotonic. What is hypotonic? When the medium around the cell is having more water, that is the dilute solution, right? Then the hypertonic solution, when the media around this cell has more water concentration, that is less water concentration, that is it is concentrated solution. And isotonic means the medium around the cell has same water as the same water concentration as that of the cell, right? So let me give you one example how this osmosis is taking place in our body, right? If you take the RBC, right? If the human RBC is taken and this is put into the isotonic solution, that is the Ringer solution. Remember this name, Ringer solution. This Ringer solution is isotonic, right? So what will happen? What will happen? This is isotonic, means this, then the cell or the RBC, this will remain normal, no change, right? But if it is hypotonic, then this will get swollen, right? Because hypotonic means the water concentration outside this cell RBC is more, so the water will enter, right? So this cell will swollen and at last this will rupture. This will rupture or hemolyzed. There will be hemolysis. structure of RBC. But if it is hypertonic, means the concentrated solution is around the RBC, is outside the RBC. So what will happen? The water from this RBC will be out, right? So this will become shrunken, or this process is called crenation. These RBCs are called crenated RBC, right? So what is the difference between the diffusion and osmosis? Just I told you, this diffusion can occur in any medium, but for osmosis, the semi-permeable membrane is required and is occurring or is possible only in the liquid medium, right? Then the diffusion molecules may be solid, liquid and gas, but this molecules must be in soluble form, right? Then the diffusion is not affected by any other molecule. But you have seen that this osmosis is greatly influenced by the other molecules. If it is more, then the osmosis, either endosmosis or exosmosis occurs. In diffusion, the equilibrium is maintained for after some time, and here the free energy of diffusion molecule is achieved, right? But in osmosis, it is never achieved. Okay? Then one more process I told you, mediated transport. In mediated transport, first diffusion, then osmosis, then mediated transport. So in mediated transport, this can be facilitated transport or active transport. So what happens, or what is the difference in facilitated transport? These are taken under the heading of mediated transport. So first is facilitated transport and second is active transport. In facilitated transport, here we will take an example of sugar and amino acid, right? That is the metabolic waste, right? So this sugar, amino acid and metabolic waste, they leave the cell by the help of a special protein that is called transport protein, or we have just seen the permease enzyme, right? So this helps actually in the transport of these substances. Now what happens? They form a small passage through which this membrane, which enables this solute to cross the phospholipid barrier, right? Now here you know that these permeases, they are very specific for every substance. So you can say that these permeases are specific, right? Then the second is active transport. Here the active transport actually needs energy, and this energy comes through the ATP pump, right? That is transmembrane ATP pump, right? Now, this transport these molecules in the direction opposite to the concentration gradient. Okay? So, there is transport of material opposite to the concentration gradient. That is why this requires energy. Now, here you remember whatever energy is stored in the ATP, about 10 to 40% of this energy is used to power this sodium potassium pump or sodium potassium transmembrane pump, right? Through this pump, this active transport occurs, right? So this was the third, right? Then the fourth we will see endocytosis. The fourth mechanism is the endocytosis. Here the plasma membrane, how it is helping us see? The ingestion of the material by the cells through this plasma membrane is known as endocytosis. Under this heading, 3 points we will discuss, that is the phagocytosis, podocytosis, right? One is phagocytosis, second is podocytosis, and the third is receptor mediated endocytosis. receptor mediated. Endocytosis. Right? Now, phagocytosis. This phagocytosis, phagos means eating, right? So this is cell eating. This you know in lower classes you have studied in the amoeba, right? This is the process by which the amoeba intakes its food, right? Then this phagocytosis is also seen in WBC of our body. This due to this process only, this enters takes the invaders or the microorganisms. The neutrophil and eosinophil, they are the phagocytes that phagocytose the foreign particles, right? And this way they are actually helping our body, right? The second is the podocytosis. This podocytosis is also called cell drinking. Cell drinking means the water comes inside, right? The unicellular cells, they are not having special organs, so this podocytosis, they take the water inside. Then the third is the receptor mediated endocytosis. For bringing the larger molecules inside, this proteins of this plasma membrane, they get adhered with these molecules, right? Then the next variety is the exocytosis. Here some vesicles, they are formed and they fuse with the plasma membrane. So in exocytosis, some vesicles are formed which get fused with the plasma membrane, and in this way they remove this content or bring this content to the outside. Okay. Then we will see in detail about the cell wall. In short, I have told you during this introduction that the cell wall is mainly present in the plant. Right? In plant there is a rigid cell wall that is present outside the plasma membrane, and this is non-living, and remember, this is freely permeable. So what I said, this is rigid, non-living and freely permeable, right? And this is secreted by the cell only. This is secreted by the cell and is responsible for maintenance of its shape. Also maintains the turgidity and prevents desiccation of the cell. Right? This prevents desiccation of the cell. Now, what is the function? If you are asked what is the function of cell wall, then you will write simply that this permits the plant cell to become turgid, then provides mechanical strength and also support, protects from the outside, right? Then this contains some pores through which the fine structures, thread-like structures are passed, and they are known as plasmodesmata, right? And this allows the exchange of material in the nearby living cell contact, right? Now this cell wall, they are also glued by middle lamellae that contains the calcium and magnesium pactate. Okay? Clear? Middle lamellae is actually helpful in the structure that helps in gluing of the cells, right? Now we will now come to a very important structure, that is the nucleus. Nucleus. This is the centrally located spherical cellular component, right? And is bounded by two nuclear membrane, right? And this forms the nuclear envelope. And between this two nuclear membrane, there is presence of nuclear space. These are the nuclear membrane that forms the nuclear envelope, right? Now, this nuclear envelope is also having small pores, right? Very small pores, they cannot be seen like this. And this encloses the ground substance that is called nucleoplasm. This nucleopores, they allow actually the transfer of materials between this nucleoplasm and cytoplasm. Right? The nucleolus and chromatin material, they are embedded in this nucleoplasm. Right? So, what is nucleolus? This is, remember that nucleus contains nucleolus, and nucleolus is actually rich in RNA and protein. This is rich in RNA and protein. So if this is rich in RNA, that means this is the factory of ribosome, right? And this ribosome, this helps in protein synthesis, right? So remember, this nucleolus is the factory of ribosome, and these ribosomes are the factory of protein synthesis, right? Then this contains the chromatin materials. They are thin finger-like intertwined masses of this chromosome material. Their genetic material is the DNA and the histone protein. Remember, these eukaryotes, they contain this histone protein, whereas these prokaryotes were lacking this, isn't it? This stores all the information from the cell to cell, right? This actually stores all the information according to which these cells act. This nucleus, which is found in the eukaryote, right? This is found in each cell of eukaryote, but it is absent or it is not present. I am writing so that you can remember this point. This is absent in the lens of eye, then IBC of the mammals. IBC of the mammals and the sieve tube of the plant. Remember, this nucleus is absent in the IBC of the mammals, lens of the eye and the sieve tube. Right? This nucleus actually contains chemically the nucleic acid and protein that I told you. So this consists of nucleic acid, that is the rDNA, and the nuclear protein, mainly the histone, right? Now this nucleic acid, that is, again I told you there are two types, the RNA and DNA, that is the ribonucleic acid and deoxyribonucleic acid. They are actually the macromolecules and polymer of the nucleotide. Now let me clear one more. Now let's see this nucleic acid in detail. I told you that this nucleic acid can be ribonucleic acid, or that means the RNA, or deoxyribonucleic acid, or DNA, right? Now here this RNA or DNA, they are the macromolecule and actually they are the polymers of nucleotides, right? They are the polymers of nucleotide, and each nucleotide, they have a 5-carbon sugar, then the nitrogenous base, and this nitrogen base is either purine and pyrimidine, right? And one phosphate group, right? So the nucleotide is made up of 5-carbon sugar, nitrogen base of purines and pyrimidines, and a phosphate group, right? So this nucleotide undergo polymerization and this forms your nucleic acid, right? But if this nucleotide, if this undergoes hydrolysis, if this nucleotide undergoes hydrolysis, then this gives nucleoside. And this nucleoside is mainly consisting of your nitrogen base and 5-carbon sugar, but this phosphate is absent. Right? Or you can say that this nucleotide on hydrolysis, this gives nucleoside and phosphate group. Right? So if you are asked to write the structure of nucleoside, then you will write this consists of nitrogen base, that is purine pyrimidine, and a pentose sugar or 5-carbon sugar. Right? Okay. Now, this RNA and DNA, if you see, they actually differ in this pentose sugar only. There is structural difference in this sugar. Right? Now, let's see what is the difference between this DNA and RNA. Right? This DNA, this contains the deoxyribose sugar. Deoxyribose sugar, and this contains ribose sugar. Okay. Then this DNA here, the hydrogen is attached to the second carbon, but here this original or this in the second carbon, what I am saying, this here hydrogen is attached, but here the hydroxyl group is attached to this second carbon. Right? Then the nitrogenous base, they are here adenine, guanine, cytosine and thymidine. Right? Whereas in this, the adenine, guanine, cytosine and uracil. Here the thymidine is present and here the uracil is present. Right? Then this DNA is actually double stranded. Right? Double stranded, or there are two chains of nucleotide. Two or double chain of nucleotide. Right? And here there is single chain of nucleotide. Single chain of nucleotide. Right? Now, if there is double chain of nucleotide, that means this is forming a helix, double helix. But this is here helix is not seen in the RNA. Okay. Now we will see in detail the structure of DNA or deoxyribonucleic acid. So this DNA structure was given by Watson and Crick. This was given by Watson and Crick. Right? This DNA molecule is I told you just now that there are two chains or double chains. So they are so they are the twisted ladder pattern. Twisted ladder pattern. Right? The upright arm that composed of, they are like this. Right? So one is upright arm. Right? And this mainly consists composed of the chains of alternate sugar, that is the, so this consists of the sugar and phosphate group alternate. Right? And the crossbars, here the crossbars, they contains the nitrogenous base. Right? So if you see, this adenine, this get bind with the thymine, and there is double hydrogen bond. But this cytosine, this combines with this G, now the guanine, that is there are triple hydrogen bond. Right? So these crossbars, they are containing this nitrogen base. Right? And the upright arms, they are composed of alternate sugar and phosphate. Clear? The structure of DNA is clear? So these crossbars, they are formed by the linking purines and pyrimidines, and the upright arm, they are formed by the alternate sugar and phosphate. And there is double hydrogen bond between adenine and thymine, or thymine and adenine, and the triple hydrogen bond between cytosine and guanine. So this is the primary structure, right? This is the primary structure, right? Then we will see the secondary structure. Secondary structure. What happens? This double-stranded helix, they are formed by coiling of the two-layer polyhydroxy ribonucleotide strand around an identical central axis. Right? And the B form of DNA is predominant form that is present in this living cell, and this B form is the actually secondary structures. Right? Here the coils of double helix, they are very regular and smoothly curved. Both ends of this helix, they form a right-handed coil, but they are anti-parallel to each other. Remember, they are anti-parallel to each other. That means 5' end of one, this faces the 3' end of the other, and vice versa. Okay? So this is 5', this is 3'. Here this is 3', this is 5'. Right? Now the two strands of this double helix, they are actually held together by the hydrogen bonds, I told you, that is present between this complementary bases of the two strands. Now each strand is again completely held by the hydrophobic force between the rings of this consecutive bases. Right? The hydrogen bond that binds the purines and pyrimidines, they they are respectively to one purine, then pyrimidine, one pyrimidine to pyrimidine, right? So they are actually, what I am saying, this hydrogen band, they bind this purine and pyrimidine residue of one strand with the respective purine or pyrimidine of the other. Remember, whenever there is bonding between this two nitrogen base of the two bands, they have to be respected, right? Means the adenine will bind with the thymine and cytosine with the guanine only, right? Now, what is the base pairing rule? Base pairing rule, I had already told you, but you write it down, that this is the base pairing rule, is that adenine will bind with thymine and there will be a double hydrogen bond, right? But the cytosine will bind with the guanine and there will be triple hydrogen bond. Regarding this, actually Chargaff was a scientist who gave a rule. Chargaff's rule. He said that the number of adenine and guanine residue, they are equal to the thymidine and cytosine residue of the two strands of the double helix, right? Then the purines must be equal to pyrimidines, right? And the two strands are complementary to each other in base pairing. This must be complementary to one another in base pairing, right? Then the distance between the two chains of DNA is, two chains of DNA are at the distance of 20 angstrom, whereas the two nucleotides, they are at the distance of 3 to 4 angstrom, right? So this is the Chargaff's rule. Okay? And according to this Chargaff's rule only there is pairing. Now, I told you that the backbone is made up of phosphodiester bond, right? 1 minute. So the phosphodiester backbone of each strand, they remain towards one side of the coil, and the outer surface is formed by the pentose phosphate residue of the nucleotide. Right? Here the pentose ring, they lie parallel. What I am saying? The pentose ring is parallel to the axis of helix. Is parallel to the axis of helix. Right? The pentose rings, they lie parallel to the axis of helix. And the polar group of the sugar and phosphate, they are mostly exposed to the surrounding aqueous. Right? The two bases of each base pair, they face each other. Non-polar rings, right? The polar rings, I told you, they are exposed to the surrounding aqueous medium, and the non-polar rings, they lie to tend to be stacked parallel to each other. Right? In this way, the hydrophobic interaction is developed between them. Right? The non-polar rings of the purines and pyrimidines, they tend to be stacked parallel to each other, and that is stabilized by the hydrophobic interaction between them. Okay? This consecutive, and there are some hydrogen bonds also, base pair, that lie in the parallel planes at the distance between these two is 34 nanometer. 34 nanometer from each other, right? And there are 10 base pairs, right? There are 10 base pairs. So the pitch of helix is 3.4 nanometer. What I am saying, the consecutive hydrogen bonded base pairs, they lie in parallel plane at the distance of 0.34 nanometer. And each turn this has 10 base pairs. So the pitch will be 3.4 nanometer. Okay. Now there are some special forms of DNA. And this I will tell you first the Z form. Z form of DNA. So this Z form of DNA, this occurs actually in the short stretches of Drosophila. The Drosophila chromosome and the human DNA, this has, what I am saying, the short stretches of Drosophila and human DNA is actually Z DNA or Z form of DNA, right? And this consists of two helix or double helix of the two linear deoxyribonucleotide strands, isn't it? Deoxyribonucleotide strand, and they are undergoing polymerization, so polydeoxyribonucleotide strands. Now each strand that carries the alternate purines and pyrimidines deoxyribonucleotide residue in the repeated sequence. And here the strand that forms the left-handed coil around the central axis of the double helix. Left-handed coil. Remember, this forms the left-handed coil around the central axis of the double helix. Each turn of this helix, this carries about 12 base pairs. This carries 12 base pairs. And the pentose phosphate pathway is actually zigzag. That is why this is called zDNA, right? Instead of having the smooth curvings, this is zigzag pattern, right? And the diameter of helix is 1.8 nanometer, right? And this exists at high ion concentration, right? Now, another forms, there are the A, C, D and E forms of DNA are also found, and they have the double helix, and this exists, they actually exist and carry about the base pair terms, the number of base pairs. If I write this A, C, D, E, right? This types of DNA are also existing, and the difference is that they carry the number of base pairs, they are different, right? So A carries 11 base pairs, C carries 9, 1 upon 3, and this D contains 8 base pairs and E contains 7.5 base pairs per turn. Remember, this is per turn, and this is containing 12 base pairs per turn. Only this is the difference in the number of nitrogen bases, right? Now, the next variety, that is mDNA or mitochondrial DNA. Mitochondrial DNA or mDNA, right? Here the primary structure is made up of 10 to the power 4 nucleotides. 10 to the power 4 nucleotides, right? And the secondary structure is double stranded, and they are endless, and they are in the closed loop or circular, right? Endless, no end, right? Now this have a supercoiled tertiary structure also. Okay, the prokaryotic bacteria, they have single chromosome and single circular DNA, and the double helix that is found is 4:10 to the power 6 base pairs. Okay. So this mitochondrial DNA is having 10 to the power 4 nucleotides, and this circular or having no end, endless, right? Now let's come to the next, that is nucleoprotein. Nucleoprotein. This nucleoprotein is actually a complex protein. They are protamines and histones. They are protamines and histones, right? And some non-acidic, they are the non-acidic, right? And the acidic protein, non-histone protein and nuclear enzymes are also present, right? So let's see the protamines. They are the single, or they can say they are very simple. They are very simple basic protein. They are very basic, simple protein, and they have low molecular weight. As they are basic, they have low molecular weight, and they are very rich in the basic amino acid. Right? Basic protein, so basic amino acid. That is example, or you can see the arginine. So this is rich in the basic amino acid arginine, right? And they are mainly found in the sperms of fish. They are mainly found in the sperms of fish, and they are tightly bound to the DNA, right? Then next is the histone. Histone is the basic protein. I had told you that protamine and histone, they are the basic protein, right? This is again a basic protein, and they are this exhibit a greater degree of heterogeneity than the protamines, right? This is basic. Why basic? Because of the high proportion of the amino acid. They are basic amino acid, that is arginine and lysine. So there is high proportion of basic amino acid arginine and lysine, right? And remember that this histone lacks tryptophan. Remember this point, that this lacks tryptophan, right? The molecular weight of this histone protein is 11,000 to 21,000. There are 5 principal histone proteins are found, or you can say the types of histone protein are 5, right? H1, H2B, right? Then H2A, H3 and H4, right? So H1, H2A, H2B, H3 and H4. These are the 5 basic types of this histone protein, right? Now, except this H1, all eukaryotes, they have the same amino acid sequence, right? What I am saying, except H1, the amino acid sequence of all these are having same amino acid sequence, and the H4 is actually, this is actually mainly conserved for the evolutionary point of view, right? So this has been conserved through evolution, right? Now, what is the role of this histone? This actually acts as repressor or inhibitor of gene, right? So this histone, this acts as repressor or inhibitor of the genes, right? Then they are made up of, or you can say that their mode of regulation of this gene transcription is in a non-selective pattern, right? And why this is non-selective? I am saying that their mode of regulation of the gene transcription is non-selective pattern because this histone lacks any tissue specificity, right? Then the interface nucleus and the chromatin, they contain the non-histone protein, and some of them, they are linked to the DNA. In this condition, they are called residual protein. Residual proteins. These are the non-histone proteins that are linked with the DNA in the interface, right? Now the prominent DNA that is linked, acidic protein, is the heterogeneous group of phosphoproteins, right? And these phosphoproteins, they are very high in number, or they are abundant in the metabolically active cells. Then I told you that this acidic protein, that the protamine and histones, they are heterogeneous and they show phylogenetic as well as tissue specificity to the DNA. I am again repeating this point. These histones and protamines, or the acidic proteins, they are heterogeneous and they show the phylogenetic as well as tissue specificity to the DNA. And they do have the stimulatory effect on the transcription because they are selectively binding to the DNA. Okay? Now, there are some nuclear enzymes also. And these nuclear enzymes are, they are DNA polymerase and RNA polymerase, then endonuclease, right? So they are DNA polymerase, RNA polymerase, endonuclease, lipase, right? So these are the nuclear enzymes which are concerned with the synthesis and repair of the nucleic acid, right? So this nuclear enzymes, DNA polymerase, RNA polymerase, endonuclease, lipase, and many, they are mainly concerned with the synthesis and repair of the nucleic acid. The enzymes that are related to the nucleoside metabolism and bound to this DNA, they are NAD synthetase. So what is the NAD synthetase? There are three enzymes: NAD synthetase, nucleoside triphosphatase, and histone acetylase. Nucleotide, what I am saying, NAD synthetase, nucleotide triphosphatase, triphosphatase and histone acetylase. These are the three enzymes. They are related to the nucleoside metabolism. Nucleoside metabolism. Nucleoside means nitrogen base and sugar, pentose sugar, right? Now, the presence of this enzyme of the glycolysis is also seen in the nucleus. So, what does it suggest? This suggests that the cell nucleus, this uses the glycolytic pathway as their main source of energy. Okay? This nucleus gets energy by the glycolytic pathway because we get the enzymes of the glycolysis inside the nucleus also. Okay. Now we will see the ultrastructure of the nucleus. That if you see the ultrastructure of the nucleus, the first is the, what do we get in the ultrastructure of the nucleus? First is the nuclear membrane, or this is also called karyotheca. Karyotheca, right? Then nucleolus or true nucleolus. So what is this? This is the ultrastructure of nucleus. Then the third, there will be nucleus sap or nucleoplasm. Then the fourth is the chromonimeta or chromatin fibers. Chromatin fibers, right? Now the nuclear envelope or the karyotheca, this is the outer envelope and is again made up of two unit membrane. This is lipoproteinous in nature, and there is a space, that is the perinuclear space between these two membrane, right? I already told you. And the perinuclear space is about 100 to 150 angstrom unit. The outer membrane, this actually often appears rough because this is associated with the ribosomes, and they may be continuous with the membrane of endoplasmic reticulum. Remember, the outer nuclear membrane, this appears rough because this is having association with the ribosomes, and this they may be continuous with the membrane of endoplasmic reticulum. Right? Then I told you that this nuclear membrane contains pores. Right? Now here you remember that this nuclear envelope is actually derivative of the membrane of endoplasmic reticulum. So this nuclear envelope is the derivative of the membrane of endoplasmic reticulum membrane. Right? So we will take the cell division in detail, but now here I would like to tell you, you notice that in telophase, this is a step in the mitosis, the cisternae of this endoplasmic reticulum, they collect around the chromosome, and by fusing together, they form the nuclear envelope. Right? During telophase, the nuclear envelope is formed. Right? Now, what is the function of nuclear membrane? This, if you see in oocyte, what I am saying, now I am telling you the function of nuclear membrane. In oocyte, this allows free exchange of ion between the nucleus and cytoplasm. Right? Then, in the salivary gland of Drosophila. In the salivary gland of Drosophila, the nuclear membrane, this acts as barrier for the diffusion of substance, and even the ions like the potassium, sodium and chloride. Right? The nucleotide, the messenger RNA, the ribosomes, they all move in and out through this nuclear pore that is present in the nuclear membrane. right? Now then here magnesium ion activated ATPase activity is also associated with the nuclear membrane as well as with the pre-complexes, right? Clear? These enzymes, whatever, they are actually responsible for the active transport of the molecule and the ions across the membrane. Okay. Then the next structure is the nucleolus. This nucleolus was discovered by Fontana. Remember, this was discovered by Fontana. This is the large eccentric spherical acidophilic nucleus. And number of nucleoli is also variable in different cells, right? This is prominent in the cell and which shows the synthetic protein or other substance having their molecules. The sperms, the muscle cells, they are having small nucleolus, right? But I am saying that this nucleoli is prominent in the cell showing synthetic proteins or other substances, right? And they are having the large nucleolus. And small nucleolus is present in the sperms and muscle cells, right? Now, this contains the phosphoprotein but no histone, okay? In nucleus, you have seen the histone is present, but in nucleolus, histone is absent. Okay, this nucleolus is also called small nucleus or plasmosome. Plasmosome. Right. Just I told you that the number of nucleoli is variable, right? And if you see in the diploid cell, they have 2 nucleoli, but the somatic cell of the human, they have 5. The oocyte of the xenopus is having 1600 nucleoli. Remember, oocyte of xenopus has 1600 nucleoli, right? Now, if you see the structure of nucleoli, this is a darkly stained granular or naked organelle, right, with no limiting membrane, and this is having 4 parts. 4 parts. This nucleoli is having 4 parts. First, pars amorpha or matrix. Pars amorpha or matrix, right? Then second, the granular zone, and this granular zone is mainly made up of ribonuclear protein. Granular zone, which is made up of ribonucleoproteinaceous fibrils, right? Then the granules, right? In the perinuclear area, right? So this the chromatin. Third is the granules, and then the chromatin. And this chromatin is again of 2 types: perinuclear chromatin and intranuclear chromatin. Perinuclear and intranuclear. Okay, so perinuclear chromatin, intranuclear chromatin, right? Next, this nucleonema and outer nuclear region, they are collectively associated with the chromatin, right? Now this is the pars amorpha, right? Then there is pars chromosoma of the nuclear DNA, and then this again differentiated in the cytosol. This is chromatin, but this is the pars chromosoma. This is also called pars chromosoma. This is having perinuclear and internuclear chromatin, right? And mainly this is made up of RNA and non-histone acidic proteins. Their origin is from nuclear DNA, right? And they help or their function is the biogenesis of the ribosomal RNA, right? And they also help during spindle formation, right? Then they combine with the ribosomal protein and forms the ribosomal subunit. Okay. Then these chromatin threads, they are nucleoprotein in nature, right? And during cell division, these chromatin fibers, they become thick and they can be clearly visible under microscope, light microscope even. And in this case, we call them chromosome. These chromosomes, they are capable of cell reproduction and they play a very vital role in the heredity, mutation, then variation and evolutionary development of the species. So let's see, I have told you two names, that is chromosome and one is chromatin, right? The two terms, right? Don't get confused with this. Chromosomes are the condensed part of the nucleoprotein complex, whereas this chromatin is the uncondensed part. This is the condensed and this is the uncondensed of the nucleoprotein complex, right? And then these are seen only in the M phase or atavic phase, right? So during the M phase of nuclear division only they are seen, and this chromatin they are seen in the interphase. Then the third difference is that these chromosoma, they are concerned with the distribution of genetic information to the daughter cell. But this chromatin, they control the metabolism and other activity in the cell. Clear? Now, the type of this chromatin, actually this chromatin, they are again depending upon the type, they take the scale, right? The staining property, they are named heterochromatic and euchromatic, right? So the heterochromatic and euchromatic, see this heterochromatin, they are darkly stained. They are darkly stained, and this euchromatin, they are lightly stained. Right? So the heterochromatin are the darkly stained. Then they are actually condensed region of the chromatin fibers, and here they are less tightly condensed. Right? They are transcriptionally inactive. This transcriptionally inactive, but they are transcriptionally active, right? Then this heterochromatin, they are not affected by any variation or less affected by any variation in the temperature, age and sex, right? But this euchromatin, they are very much affected, or you can say more affected. They affected more and they are less affected. Here we must write less affected by the temperature, age and sex, right? Now the frequency of crossing over is frequency is low, whereas here the crossing over frequency is high, right? Then here they are the replicability is late, right? So they shows late replication and here they show early replication. Right? So this is the difference between the heterochromatin and euchromatin. Right? Now the chromosomal number, you know that this is fixed for a species. Chromosomal number is fixed in a species. So let's see the number of chromosome, highest number of chromosome and lowest number of chromosome is found in which animal? The lowest number of chromosome is found in Ascaris, and this is 12. This is 12, right? And the highest is found in Ophioglossum. Ophioglossum. And this is 1262. Right? And the size also varies, but they are constant for a species. Right? This chromosome actually contains a clear zone that is known as, the clear zone is known as centromere. Let's see the structure. I am telling you the structure. This is having a central clear space, the clear zone, that is the centromere or kinetochore. Right? And then the centromere, this is divisible into chromosome up to 2 parts. This is chromosomal arm. This actually divides, the centromere divides the chromosome into 2 parts, and each part is known as chromosome arm. Right? And the position of the centromere, this also varies. Right? And depending upon the position of the centromere, the chromosomes are now we will see the types of chromosome depending upon the position of centromere. Right? First is the telocentric chromosome. Here, this centromere, this occupies the terminal position. Then this is your telocentric. So here only one arm is present. Right? Then second is acrocentric. Acrocentric means here this is present in such a way that one arm is long and one is small. Right? So the centromere is at one end. Right? This is at one end. Therefore, the two arms are unequal. Right? One is exceptionally long and another is short. Right? Then the third is submetacentric. Here they actually form the J or L shape. Right? The position of centromere is such that this forms the chromosome J or L shape. Right? Then, so this is submetacentric. Then metacentric. Here the centromere is such located that this forms V shape. Right? So this forms a V-shaped centromere in the center, and the two arms are again equal. Right? So depending upon the position of the centromere, these are the types of chromosome. Right? This type and shape of the chromosome is determined by the primary constriction. Now let me first draw a diagram. So that this will be more clear to you. See, this is the pellicle, this is the part known as satellite, this is the promonimeter, this is the secondary constriction, the matrix, the kinetophore, the primary constriction, secondary constriction. Okay, so this I am saying that the type and shape of the chromosome is actually determined by the position of this primary constriction, and this is located at the point where the arms of the chromosome they meet, right? Now within this constriction a clear zone is indicated where these small granules or spherules are present. Right, and this clear zone is the centromere or kinetophore. This is the centromere or kinetophore. So what is this? This is the zone or clear zone in primary constriction, right? Primary constriction where they contains the spherules or the granules. Now this clear zone is actually or the position of this clear zone or kinetophore or centromere is constant for a particular chromosome. Remember, the position of this kinetophore or centromere is constant for a particular chromosome. Most of these chromosomes are monocentric, means one centromere, but they may be di or polycentric chromosomes. Okay. The chromosomes having, they also have, besides this primary constriction, this also had the secondary constriction, and this is considered as nucleolar organizer. Nucleolar organizer. So the secondary constriction is also considered as nucleolar organizer. Now this position is constant, remember. So in man, the secondary constriction that is found is on the long arm of, we must know about the man, right? Here in man, this secondary constrictions are present over the 1, 10, 13, 16 and Y chromosome, right? Why this is called nucleolar organizer? Because this is necessary for the formation of nucleolus, right? Now the part of chromosome beyond this secondary constriction is very short, and this appears as a small sphere that is called satellite. Satellite, right? So the chromosome that bears this satellite is known as SAT chromosome, right? SAT. What is SAT? The chromosome with this is known as SAT chromosome. SAT is sine acid thymonucleononic, means without nucleic acid. This is the portion without nucleic acid. So this is sine acid thymonucleinico. Sine acid thymonucleinico means the nucleic acid is acid, right? The structure is clear. Now here the tip of the chromosome is known as telomere, and this has unique property that this prevents the end of the chromosome to stick from each other, right? So these telomeres, they are actually specified regions of the chromosomes for attachment of nuclear envelope. This prevents them to stick to each other and also is the site of attachment of nuclear envelope, right?